Hi everybody, I'm Edward, working as Assistant Professor in the Department of Microbiology, St. Mary's College, Autonomous Thuthukudi. I'm going to discuss about mRNA structure and its function. Where the mRNA fits, the central dogma. The DNA is transcribed into mRNA and mRNA is translated into protein. So it is transcribed from DNA in the nucleus, then travels to the cytoplasm where ribosomes translate it into protein. What is mRNA? It is a single-stranded ribonucleic acid molecule. It carries genetic information copied from DNA in the form of series of three nucleotides codons to direct protein synthesis. It is single-stranded. It has ribose sugar. It uses uracil and it is short-lived. This is the anatomy of mRNA transcript. It has four important parts. One is FIDAS cap, UTRS, coding sequence, and poly A tail. The FIDAS cap. It is a modified 7-methyl guanosine nucleotide joined by an unusual triphosphate linkage to the first base of the transcript. It protects the transcript, flags it for export, recruits the ribosome, aids splicing. And untranslated regions, there are two untranslated regions, FIDAS and UTR. It contains sequences and structures that help ribosome to locate the start codon. And 3-UTR binding site for microRNAs that fine-tune mRNA stability and translation with other features. This is a coding sequence and the triplet code, an unbroken run of codons, each a group of three nucleotides read by ribosome in one direction, one codon at a time. The poly A tail, it's a chain of roughly 150 to 250 adenine nucleotides added enzymatically to the 3-N. It guards against decay, assists nuclear export, boosts translation, shrinks with age. This is the process of mRNA development. It starts with initiation, elongation, FIDAS capping, splicing, and termination of poly A. This is a translation part. It starts with initiation, elongation, and termination. The key players are ribosome, tRNA, and mRNA. Why mRNA matters beyond textbook? Structural parts engineered for stability and potential in mRNA vaccines, modified nucleosides in immune overreaction. Same platform can be designed for new targets, changing only the coding. This is a summary of mRNA. First one is structure. Structure starts with 5' cap, 5' UTR, coding sequence, 3' UTR, and poly A tail. Origin transferred from DNA in the nucleus by RNA polymerase 2. Job to carry the genetic message to ribosome for translation into protein. Control point, every structural feature is also a regulatory lever over gene expression. Real impact, the same principles underpin modern mRNA vaccine and therapeutic design.